Hello everybody and welcome back. So, today we will be on module 10. In the last module, that was module 9, I have mainly discussed on the biomolecular probes, basically the constructions of artificially synthesized molecules that you can use to study what is going on inside the cells. In other words, that will be giving you reports back to you. So, detection purpose, to see the live cell imaging where your biomolecules are actually going and what is happening. Those, in order to know that, we have synthesized, or nowadays lot of different kinds of artificially synthesized molecular probes have been used. So, I had tried to give you a kind of one or two important ones of them, such as PNA and LNA along with siRNA, which can act as biomolecules, especially for research purpose. So, today we will be talking about a different topic, that is chemistry of carbohydrates. So, carbohydrate is, of course, you know that it is one of the very important biomolecule that is present in our body, in our cells. Carbohydrates constitute a major part of our body constructions, and it is also a major source of our cellular energy. So, there are different kinds of carbohydrates that are present in animal body, in plants, in microorganisms, such as if you talk about the plants, the stems of many different plants such as bamboo, straw, wheat, a majority of them are basically constructed of carbohydrates. And obviously, the small carbohydrate molecules, the small molecules containing the sugars and other kinds of carbohydrates, are the major source of the energy in plants. Similarly, in animals, in higher organisms also, cellular membranes are constructed of glycolipids. So, it is basically a combination of lipids and carbohydrate. Also, there are other parts of the body that are that have carbohydrates in it, and carbohydrates metabolism is obviously a major source of our energy. Microorganisms also have the same things, and apart from those, there are other kinds of small molecules. Of course, they are also biomolecules, and they are present in our body, in higher organisms as well as lower organisms, and that have carbohydrate part in it or sugars part in it, such as obviously we have seen before to a large extent a DNA that is constructed of the deoxyribose sugar, RNA that is constructed of D-ribose sugar. So, these are the constructions of DNA and RNA on large biological macromolecules. Small molecules includes ATP, adenosine triphosphate, adenosine diphosphate. They are the source of energy in our body, the cells. And obviously, along with that, you know, there are many coenzymes, there are many enzymes that have glycosidic part in it. There are many coenzymes such as NADP, nicotinamide adenine dinucleotide phosphate, FAD, flavin adenine dinucleotide, and so on. There are many others, and all of these coenzymes have sugar unit attached to it. Most of the times it is the ribose or the deoxyribose. So, these are the smaller organic molecules or smaller biomolecules that contain the carbohydrates. Here, if you see, lot of drugs, some of them are natural products, you can isolate it from natural sources. Some of them are synthesized also. They are very popular, very well known drugs. These are mostly the anti-tumor agents. Doxorubicin is a very famous chemotherapeutic agent which is used in many different types of cancers. It has a glycosidic bond and that has a carbohydrate or the sugar moiety which is little bit modified of course here. Doxorubicin was initially synthesized from donorubicin. Donorubicin is a naturally occurring molecule or is a natural product. So, this has the amine group and this is very close similarity with the doxorubicin structure. This is epirubicin, this is idarubicin. So, many of them are available as drugs in the market and they contain the carbohydrates. So, in general carbohydrates basically they are the organic molecules or organic compounds that show biological applications Not only that, they are themselves are biomolecules. So, they are the molecules that are abundant in the biological system. As I have said, they are the major source of energy. Also, carbohydrate is the major source of the carbon that are needed in various cellular activities or to synthesize various molecules in the cells, carbon containing molecules. It is a major source of carbon in cells. Obviously, most of the smaller molecules that are present in cells have carbons in it, and many of those molecules or the biomolecules are actually derivatized or actually synthesized back from carbohydrates. So, it is a major source of carbons in the cells also. They also serve as storage unit in biological bodies. Or biological cells. Now, the name carbohydrate basically means that if you char a molecule, what you get is carbon and hydrates, means water. So, that is where the name came from. Carbohydrates means it produces carbon plus water. When you make it to undergo oxidations, massive oxidations. So, it is a major source of energy because that is due to the metabolism effect, which basically means the oxidation of the carbohydrates that produces the energy. Now, carbohydrates can be divided into many different kinds. So, carbohydrates have the types of carbohydrates. There are many different types. One is of course we call it monosaccharides. Carbohydrates are also known as saccharides. Monosaccharides. Monosaccharides are the carbohydrates which has a single carbohydrate unit, such as obviously glucose, fructose, ribose of course, and all sorts of smaller structures which has a single carbohydrate unit is called monosaccharide. Of course, I think most of these things that I will be around the carbohydrates and their structures, their utilities, most of those things you already know of, but for the sake of the course I think I will go through them again. So, monosaccharides, second division is called the disaccharides. Disaccharides are the molecules, so having a single carbohydrate unit. Disaccharides have two carbohydrates units, carbohydrate units, and those are connected by glycosidic bonds. Connected by glycosidic bonds. A single glycosidic bond would be there. So, it is a bond. The example of such the disaccharides can be lactose, sucrose, and so on. I will show you the structure of lactose, for example. If you draw the chair conformation, this is O, CH2OH, OH, this is the equatorial, has hydrogen in it, and here the beta ionomer down the plane, this is up the plane OH. Now, this is the glycosidic bond O, and there is another one that can be developed from here. So, is this O, this is the other one. Here is your hydroxyl group, this is OH, OH and beta OH. So, you have a single glycosidic bond here, and this is the structure of lactose, which has two of the carbohydrate units. So, it is a disaccharide. The third one is called oligosaccharide. Oligosaccharides, obviously the name itself represents that it should have a multiple number of or more than two number of carbohydrate units. So, in this case typically the oligosaccharides are the carbohydrates that contain around 3 to 10 units of monosaccharides. So, typical examples are dextrins, dextrins are an important class of oligosaccharides. They have lot of use in, pharmaceutical industry, in textile industries, in other industries as well. So, lot of industrial applications from dextrins. Other one is the raffine series, raffinose or raffinoses series of molecules. They contain multiple number of less than 10 number of monosaccharide units attached via the glycosidic bonds together. That is called the oligosaccharides. And the fourth one, which you know a lot of examples of, are called polysaccharides. Polysaccharides are basically the polymers of the monosaccharides. Polymers of the monosaccharides. More than 10. More than 10 units connected together via the glycosidic bonds. And these monosaccharides can be different. Different means in the same polysaccharide there can be multiple number of multiple kinds of monosaccharides. Multiple number of course is there, along with that multiple kinds of monosaccharides can be connected together to give you the polysaccharides. Cellulose, present in many plants, is a major material that is obtained from plants. Glycogen, starch, these are all part of the polysaccharides. They have different kinds of units that are connected together to give you the polymeric structure, and that is why they have very long structures, very tough structures sometimes. So, I will give you a table regarding the division of these sugars, and then we will move on to the chemical part. So, if you make a table, so this is the class, what kind of sugar, this is subgroup, this is the components or the what materials are present in them. Sugar can be monosaccharide, examples are glucose, galactose, fructose, xylose. I will show you all the structures also just a little bit later. And then you have the disaccharide, as we have talked. Examples of the disaccharides are sucrose, lactose, trehalose. Trehalose is a very important carbohydrate or a very important sugar containing molecule which has lot of biological applications. Maltose, and then polyols. Such as very well known sorbitol, mannitol, so that is one. So, basically first class sugar means which has 1 unit of monosaccharides or 2 units of monosaccharides. disaccharide, and then comes the oligosaccharides. It's the first, second is oligo, saccharides. The example I have seen given is dextrine. It can be in general maltodextrines. So, oligosaccharides would be between 3 to 9 numbers of monosaccharide units. Then fructo oligosaccharide is also there. And raphins, and the last one is, so this is basically 3 to 10 units. Then comes the polysaccharides, polysaccharides are more than 10. So, more than 10 units of sugars. They can be divided into two parts. One is the starch containing, amylose, and then amylopectin. And then non-starch. Such as glycogen, cellulose of course, pectins, and so on. There are other examples also. So, these are briefly the divisions that you can make or the different classes of carbohydrates. Now, coming to the chemical structures. So, most of them of course, is a kind of have been evolved from the basic unit, which is glyceraldehyde. So, structural similarity or is from the glyceraldehyde. Glyceraldehyde has this structure OH, this CH2OH. This is glyceraldehyde, and according to the nomenclature, obviously glyceraldehyde has a chiral center here, because all 4 groups attached to the carbon are different. So, it is a chiral center which is optically active, and therefore, it should have 2 isomers. One is this, other one is this. I have drawn the Fischer projection, this here H, CH2H. This is also glyceraldehyde. So, according to nomenclature, this is D-glyceraldehyde, because the OH is on the right hand side, dextro. So, D-glyceraldehyde. So, now here in this class I will talk about only the monosaccharides, and all those monosaccharides are mostly of D configurations. So, and this is L-glyceraldehyde. Glyceraldehyde. So, I will not talk about the L isomer, we will mostly talk about the D isomer. Now, D-glyceraldehyde, if you go 1 carbon up from here, then what you synthesize, what you get is 2 here, 2 optical centers or 2 carbon chiral centers. CHO, this one remain as it is D, because that is where the name is coming from. Now, here the next one can have the same, or this remains D Here this can be the opposite, OH, H, CH2OH. Now, if you have multiple carbon centers, did you hear the name erythro and trio? So, same side and the opposite side. This is known as, it basically erythro and trio, the concept basically came from the sugars. This is called erythros, 4 carbon units, both the hydroxyl groups on the same side. This is called trios, where the hydroxyl groups are on the opposite sides. So, this is the 4 carbon units, and now moving on. Erythros, this is your trios, if you move to 1 carbon up again. So, here it is basically C3, 3 carbon center, this basically becomes C4, 4 carbon centers, C4, C4, and from here one step up, ascending series 5 carbon, CHO, OH, H, OH, H. So, this should be CH2OH. Now, the new one can be OH in this direction. This is your ribose. So, the difference is in the new one. CHO, this can be the other way around. HOH remains same. So, we are adding up. So, this is what is the new addition, right. From here the new carbon is joining here. So, this should change, the others should remain intact. This is your ribose, this is your arabinose. Similarly, from trios you can draw 2. So, this is your C5, that is evolving from erythros. From trios is CHO, this is the new one. So, it can be OH, H, H, OH, OH, H, CH2OH, that is your xylose and HOH, HOH, OHH, this is your lixose. So, ribose is a sugar that is abundant for in DNA and RNA. Similarly, if you move up, I am not drawing them, you will have altrose and allose, that is the 6 membered ring with the 6 carbon, and from arabinose, that I will draw in the next page. What you will have is, because these are the most well known one, glucose and mannose. From xylose you can have again glucose also and idose. Lixose you will have galactose and talose. Those are for the 6 carbons. So, structure of glucose is very important. So, these are all aldose sugars. I will draw the structure of glucose: CHOH, HOH, this is OHH, HOH, OHH, OHH and CH2OH. This is your D glucose. It's basic sugar, a source of energy. So, it is a purely optically active molecule which shows the specific optical rotations, and this is of course aldose sugar. So, monosaccharides can be of 2 different kinds. One is aldose, means which has an aldehyde group here, and other one is the ketose such as the fructose. So, as we move on I will show you the structure of fructose. So, aldose and that would be ketose, because that has the general structure of the ketose is the first one will have the CH2OH and then you will have the keto and then the other way around it will move on. When you form a disaccharide then you need to form a glycosidic bond, right. So, I will show you first a reaction through which a glycosidic bond can be formed in a synthetic way. The name reaction is Koenig-Knorr reaction, actually KO umlaut as a German pronunciation. If you write in English you will find this spelling in book. Koenig-Knorr reaction. For the formation of glycosidic bond. In this case, if you start, for example, if you take the chair conformation, you start with a halide alpha 1 bromo. You have here CH2OH, this is OH, this is OH and this is alpha OH. So, first thing is you have to do the protection of all the hydroxyl groups, otherwise it will react, and that can be done very well, that you know already. We have done some sugar chemistry during DNA synthesis. Acetic anhydride if you add, then or acetic acid also, then this will protect all the hydroxyl groups. Bromine. So, this will be converted into OAC, this would be OAC, O acetate, CH2OAC. So, you have to form a glycosidic bond over here. It is usually done by using Ag2CO3, a mild reagent actually, silver carbonate. What it does is it will precipitate one molecule of AgBr. AgBr would be forming and that will precipitate out. That is the driving force for the reaction. So, it will drag the Br. I will show you the structure in a structural orientation. This O O Ag. This is the structure of Ag2CO3. Br minus goes there, it will take up, this will become a kind of a minus. Since the Br minus is getting out, this will create a positive charge on this carbon, that will be to some extent compensated by the lone pair of oxygen, and you will form plus this complex. OAC, OAC, OAC. Now, you have acetate and that has double bond oxygen. So, this will attack here, open this up, that will give you a better complex or better intermediate, energetically more favorable intermediate. So, basically O, here it will form a complexation, and this will be delta plus, of course, CH3. This will be positive charge over here, OAC, OAC, OAC. So, that blocks the down the plane. And now, you have to form the glycosidic bond, you react it with, just for example, you treat it with methanol. So, your first reaction is basically treatment of this compound with acetic anhydride, Ag2CO3 and methanol. So, what this will do? This will come, attack this carbon, this will open up, but this attack has to come from the top position here, because the down is blocked by the acetate. So, that triggers the stereochemistry. So, it will be up the plane, O CH3, up the plane, and then rest will come as it is, OAC back, OAC, this is OAC, CH2, OAC, and for ester hydrolysis you can cleave the acetate and get back your hydroxyls back. So, this is the formation of the glycosidic bond. So, one of the reactions which you can use to form the glycosidic bond. As I had shown that from glyceraldehyde you can approach or you can synthesize all the higher orders of the monosaccharides. So, I will show you couple of reactions which or methods which are used. This is pretty old reactions actually, many of you already know of all these reactions. Those are used to increase the number of carbons on the glyceraldehydes or on the sugars. So, one of them, so this is called ascending series, I will call them sugar ascending series. So, if you start from let us say arabinose and you want to get synthesize glucose from there, this is the structure. One, two, three. So, D, and this is D means the minus actually, this comes from your experiment. This is D-arabinose. The question is how can you increase the one carbon here. So, the reaction is called as Killiani reaction. very well known reaction, it is a named reaction, very old reaction. The way to do is to add up one cyanide over here, because cyanide is a source of carbon. So, if you treat this with KCN or HCN, not a nice reaction, because cyanides are very harmful, of course all of you know, but in a controlled way. Nowadays you can bypass these reagents in little bit modern reagents, but still. So, this is the I am talking about the old method, the actual reaction that people have used. So, if you have cyanide, if you use cyanide, what it will do? This will, cyanide is basically Cn minus, nucleophilic addition on the aldehyde. So, you will have Cn here, Cn, and this becomes O minus CHOH. It can be this way, it can be the other way around also, and rest will be as it is HOH, OHH, OHH, CH2OH, plus you will have the other one, this is H, this is O minus, HOH, OHH, OHH, CH2OH. It turned out that this is actually the minor product, and this is actually your major product. This is according to Cramp's rule, if you remember. This is a very famous carbonyl compound reactions, of which way the incoming nucleophile will come depends, that is governed by a rule known as the Cramp's rule. I will write it here. So, according to Cramp's rule, this should be the major product, and obviously O minus would become a OH. So, what you basically have now is this as a major product. You have a cyanide and you have HOH, HOH, OHH, OHH, CH2OH. Now, you have to convert this into an aldehyde. You can use it using the catalytic hydrogenation, hydrogen palladium, or if you do one electron, you can also use dibal, it's aluminum reagent, diisopropyl butyl, diisobutyl aluminum chloride. So, that will give you the basically CH2NH2, and this on further hydrolysis. So, this is number 1. Number 2 would be further hydrolysis, that will move it to the aldehyde. HOH, HOH, OHH, OHH, CH2OH. This becomes your HOH, HOH. This is actually your mannose, and from the other one, if you do this minor product, this will give you your glucose. So, if you want to have your glucose, I think you have to start with the ribose as a major product. If you want glucose as the major product, you have to start with the ribose, not arabinose. So, this is one reaction for increasing the carbon in the sugar. Another reaction is Neff carbonyl reaction. So, in general you have a CHO. I am not writing the stereochemistry anymore, just taking a general example. CH3, CH2OH. If you treat this with a nitro compound, so the idea is you have to create a carbanion that will act as a nucleophile to attack on the aldehyde. So, you have to use a base OH minus containing, or I will simply write base. It can be many different kinds of bases can be used. So, base that will actually produce a carbanion CH2NO2. These carbanions, because of the presence of the NO2 which has strong electron withdrawing ability, is quite stable, and it is a good chemistry, actually good carbanion chemistry. So, this will react here, opening up. So, you have CH2NO2. In the next phase you have CHOH and the other CHOH. So, you have created a new CH2OH, and then on base catalyzed hydrolysis this you can convert it into the aldehyde CHO, CHOH, CH2OH. So, you have now one carbon up. So, these are some of the very well known techniques to all organic chemistry methods that we use in the laboratory to vary the number of carbons. So, in this case I have talked about the ascending series, means the addition of one carbon there. Now, coming to the descending series. So, starting with higher order, how can you reduce one? For example, you start with glucose, one, 2, 3, 4, 5, 6, yes. So, this is your D glucose. If you treat this with hydroxylamine, then what do you get? This act as a nucleophile, this comes back. So, you will have CH, and then it basically liberates water, one molecule of, it is a condensation reaction, right. Carbonyl with amine condensation is very popular. So, this will have your CH double bond NOH, OHH, HOH, OHH, CH2OH. Now, if you heat this up, ideally this can remove another molecule of water, but this does not happen like that. It works best if you use a little bit of acetic anhydride. So, that will basically eliminate another molecule of water, and this will create your cyanide CN. At the same time, since you have used the acetic anhydride, it will protect all the hydroxyl groups as well. So, you will have now OAC OACH CH2 OAC. Now, hydrolysis, ester hydrolysis basically, to deprotect all this, that will create CHOH and all the rest of it. I am writing this because I need this HOH, OHH, OHH, CH2OH. If you treat this with a base, what happens? Base will take up this proton, it will go and your cyanide will be eliminated, because cyanide is also a good leaving group. So, that will expose the CHO, one CHO here, and this one carbon would be gone. So, you will have CHO and then the rest of it, one carbon down, HOH, OHOH, OHH, CH2OH. So, you have descended one carbon here. Now, this base is actually, you should not use a very strong base obviously, because this is kind of anomeric one. So, it is very labile. You need a mild base, and the reagent is you use silver nitrate in, it is called ammoniacal silver nitrate, right, in ammonium hydroxide, which is known as the ammoniacal silver nitrate. This basically generates ammonia complex plus OH minus. So, mild base. So, you have free OH minus that will act as the base here. So, this is basically the OH minus that will take up the proton, will do this chemistry, and this reaction is known as the Hol's method. To reduce sugars, okay, one to reduce a carbon. Second reaction is, if I start with the same glucose, OHH, HOH, CH2OH, glucose again, and then you treat this with bromine water. Bromine water is used to oxidize aldehyde to carboxylic acid. So, you will have a carboxylic acid and all the rest of it. Then treat this with calcium hydroxide and heat it up. You will form CO whole to Ca. CH2OH. This, if you treat it with Fenton's reagent, Fenton's reagent is basically your FeSO4 in hydrogen peroxide. Then you get elimination of carbon dioxide, and this will be, next carbon would be exposed. So, this will become obviously a CHO, and you have this OHH, OHH, CH2OH. This is another method through which you can reduce one carbon on the sugar, and this reaction is known as rough synthesis or rough reaction. Another very interesting thing is a very important reaction that a sugar undergoes, and that is for the formation of oxazone. It is called the Amadori rearrangement. If you take either a aldose or a ketose, and if you treat it with phenylhydrazine, you will get the formation of oxazone. People have used that chemistry for various other aspects also. So, I will show you the Amadori rearrangement first, and then we can maybe use it later. Aldohexose, if you start with 6 carbon with aldehyde, is called aldohexose. Phenylhydrazine NH2. Phenylhydrazine reacts with a carbonyl compound to give you phenylhydrazone minus water. It is condensation reaction, it is very well known. In NH-PH. This. Now, this can happen, and you can use a little bit of base also. Is NaHPH, this will create this OH, CHOH whole 3 OH. Now, this is the driving force, this will come and this will take up the proton and this will open up. This is basically the Amadori rearrangement. This is the rearrangement. So, you will remain with CH double bond. So, this will be here NH, and you will form a keto. Whole 3 CH2OH. So, I will basically write it here, this is coming here, it will take up, it will go there. And you exposed now another ketone. If you treat this with another molecule of phenylhydrazine, that will again condense with the keto. NH C double bond N NH PH whole 3 CH2OH. Another molecule of phenylhydrazine will hydrolyze this and will replace this by the same compound. N NH PH N NH PH CH whole 3 CH2OH. This would be formed, and this is known as Osazone formation. This actually is used, some of this chemistry has been used for, in the next one I will show you, the epimerization. If you want to change the stereochemistry of the anomeric carbon, this is one of the way to do it. Even from allose to ketose conversion, you can also do using this kind of chemistry. Thank you.